Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. About 75% of deep-sea animals produce their own light. Picture yourself descending through a water column where every additional meter strips away another slice of the light spectrum. Red disappears first, then orange, yellow, green. Below 200 meters, only shades of blue remain, and shortly after, total darkness. In that environment, organisms generate light internally through a highly precise chemical reaction. Bioluminescence is particularly widespread among marine animals, especially in the deep sea. The proportion of bioluminescent animals is remarkably consistent throughout the entire water column, from sea level down to depths exceeding 3,600 meters. Biological light is the dominant language of Earth's largest continuous habitat. Picture a city with no power at 3 in the morning. No streetlights, no lit windows, no moon visible through thick cloud cover. That level of darkness still doesn't come close to what exists below 1,000 meters of ocean. Down there, not a single solar photon penetrates. Pressure reaches hundreds of atmospheres. Temperature hovers just 1 degree above zero. Organisms living in that environment have never had any contact with sunlight, and yet they light themselves up. Bioluminescence in that zone isn't decorative. It's functional and precise, shaped over hundreds of millions of years in environments where seeing or being seen is the difference between life and death. In the abyss, any photon emitted instantly becomes a signal. With no ambient light competing in the background, a fish that fires a blue-green pulse in an absolutely pitch-black environment is sending a crystal-clear message. It works like whispering in a completely silent room. Even the smallest sound travels through the space without interference. Bioluminescence operates by the same logic, a clean signal on a noise-free channel. That's why the uniformity of luminous species throughout the water column is so striking. Where sunlight fades out, biological light progressively takes over as the main form of communication among organisms living at those depths. The distribution of this ability across such wildly different animal groups reveals something beyond localized adaptation. Bacteria, worms, jellyfish, crustaceans, squid, and fish. Organisms separated by hundreds of millions of years of history, all independently developed versions of the same light-producing tool. When a solution shows up independently across unrelated lineages, the message is clear. That solution solves a real problem so efficiently that it's been rediscovered over and over again. Biological light isn't a niche curiosity. It's a fundamental response to the pressures the deep ocean imposes on any organism trying to survive in it. In the first 200 meters, the photic zone, sunlight still penetrates with enough intensity to sustain photosynthesis. Below that, between 200 and 1,000 meters, the mesopelagic zone begins, where sunlight becomes progressively scarce. There, bioluminescence gains real competitive relevance. Predators use counterillumination, producing light on their bellies to match the faint glow coming from above and become invisible to anyone looking up from below. Prey fire off flashes to disorient attackers. Mates use specific light patterns for species recognition in an environment where close-range vision is all there is. Photoreceptors in the eyes of deep-sea fish are often hypersensitive, tuned to detect the smallest possible amounts of light. Some have tubular eyes pointed upward, scanning the residual glow filtering down from above to spot prey silhouettes. Others have retinas packed with rod cells, cells that respond to individual photons. In that context, a single bioluminescent pulse in an absolutely dark environment is impossible to ignore. The extreme sensitivity of the receptors makes light signals even more effective. Weak flashes are enough. An organism doesn't need to burn energy producing a burst of light. One controlled pulse is sufficient for the receiver to decode the message loud and clear. Unlike artificial light sources, bioluminescence is produced inside living cells and is continuously renewable. It doesn't rely on batteries, requires no outside maintenance, and doesn't heat the surrounding tissue. Engineers developing high-efficiency LEDs are chasing exactly what marine organisms have already achieved: near-total conversion of chemical energy into visible photons with minimal residual heat. Studying how different marine organisms build, control, and modulate that ability offers direct inspiration for lighting technologies, biosensors, and medical diagnostics. Nature solved that problem long before humanity ever developed transistors. Dinoflagellates are microscopic organisms, single-celled protists, that produce bioluminescence in response to mechanical disturbance. When waves or currents agitate the water, they emit brilliant blue flashes. Beaches that glow at night when the waves break owe that spectacle to these organisms. Each cell detects mechanical stress on its membrane, triggers an internal biochemical cascade, and emits a photon within milliseconds. At high densities, millions of cells firing simultaneously create an effect visible to the naked eye. This mechanical stress activation mechanism differs from the sophisticated neural control found in vertebrates, but both arrive at the same result: light produced at the right moment, in response to a specific context. Recent research maps exactly where inside each organism's body bioluminescence is produced, and which structures control its activation. In squid photophores, light-emitting organs made up of a lens, reflector, and photogenic cells, are distributed across the ventral surface in patterns specific to each species. Each photophore can be activated independently, allowing the animal to modulate the intensity, duration, and sequence of its pulses. This turns the body surface into a high-resolution signaling display. Some researchers compare that level of control to a pixelated screen, each dot activatable separately, capable of composing complex patterns in real time. The study of marine bioluminescence has accelerated over the past few decades thanks to unmanned submersible vehicles capable of operating at great depths with cameras sensitive enough to capture faint emissions in real time. Recent expeditions have documented previously unknown species using light in unexpected ways. Sharks emitting skin patterns visible only to members of their own species. Shrimp firing luminescent clouds as a countermeasure against predators. Jellyfish pulsing in rhythmic sequences whose function hasn't been fully figured out yet. Each discovery expands the inventory of solutions marine organisms have developed for surviving, using their own light in the most extensive and least illuminated habitat on Earth. To understand how a shrimp, a jellyfish, or a fish produces light, you need to picture two molecular players working together inside a cell. The first is called luciferin, an organic molecule that acts as the fuel for the reaction. The second is luciferase, an enzyme, meaning a protein that acts as a catalyst, speeding up and organizing the reaction. When luciferin combines with oxygen in the presence of luciferase, it undergoes oxidation and releases energy in the form of light. The process is remarkably efficient. Almost all the energy converts to light rather than heat, making it a cold light reaction. An incandescent light bulb converts about 5% of electrical energy into visible light and wastes the rest as heat. Bioluminescence converts virtually 100% of chemical energy into photons. The luciferase enzyme is recycled after the reaction and can be reused. No residual heat, no waste. and no physical signal that gives the organism's presence away to its surroundings. Oxidation is a fundamental chemical process. It happens when a molecule loses electrons while interacting with another, often oxygen. Rust on metal is slow oxidation. Burning wood is fast oxidation. Bioluminescence is controlled oxidation, mediated by an enzyme confined to a specific cellular compartment, tuned to release energy precisely within the visible spectrum. Luciferin doesn't burn. It gets molecularly rearranged in a precise way. Electrons excited during the reaction return to a lower energy state and release the difference exactly as photons of light. The enzyme that organizes this process determines which wavelength will be emitted, and therefore what color will be produced. Luciferins aren't all the same. Different groups of organisms use structurally distinct molecules as their luminescent substrate. Coelenterrazine is the most widely distributed luciferin in the ocean, present in jellyfish, squid, shrimp, and fish from multiple unrelated lineages. Its abundance suggests that marine organisms frequently acquire it through feeding, absorbing it from prey that already carry it, rather than synthesizing it from scratch. Other organisms produce their own luciferins, molecular structures unique to their lineage. This chemical diversity indicates that bioluminescence arose independently in distinct groups, each developing its own version of the same fundamental mechanism. Luciferase is a protein, a chain of amino acids folded into a specific three-dimensional structure that determines its function. Within that structure, there's a cavity called the active site, where luciferin fits with geometric precision. That fit positions the molecule exactly where oxidation needs to occur. The enzyme is not consumed during the reaction. It stays intact, ready to process a new luciferin molecule. This catalytic behavior is common to all enzymes. They speed up reactions without being destroyed by them. The practical consequence is that small amounts of luciferase can process large amounts of luciferin over time, making the system highly efficient in terms of the cellular resources invested. The color of the emitted light depends on the molecular structure of the luciferin and the chemical environment inside the luciferase's active site. Most marine organisms emit blue or green, wavelengths that propagate with less attenuation in seawater. Red propagates poorly underwater. Blue travels much greater distances before being absorbed. Organisms that need their light detected at a distance preferentially emit blue. Some, however, emit red, a wavelength that potential predators in the abyss can't detect, making that light invisible to most other organisms and useful only to the emitter itself for illuminating prey without giving away its position. Controlling when light is produced requires regulatory mechanisms beyond simply having luciferin and luciferase present. Cells need to keep the two components separated when the reaction isn't wanted and bring them together when the light signal needs to fire. In some organisms, luciferin is stored in vesicles, small membrane-bound compartments inside the cell, released only upon nerve or hormonal stimulation. In others, luciferase stays inactive until a specific chemical modification activates it. More complex systems involve additional regulatory proteins that modulate the intensity and duration of the light pulse with considerable precision, allowing the organism to adjust the signal to whatever the situation calls for. Green fluorescent protein, GFP, was originally isolated from the jellyfish Equoria victoria. It doesn't produce light directly, but absorbs the blue light emitted by the bioluminescent reaction and re-emits it as green. This process of absorbing and re-emitting at a different wavelength is called fluorescence. GFP became one of the most important tools in modern molecular biology. Researchers genetically fuse it to other proteins of interest, making it possible to see under a fluorescence microscope where and when those proteins appear inside living cells. 3 scientists who developed this application received the Nobel Prize in Chemistry in 2008. Marine bioluminescence, therefore, provided a central instrument of contemporary cell biology. Bioluminescent bacteria deserve a separate mention. Species of the genus Vibrio colonize the interior of light organs in certain fish, and squid in a symbiotic relationship. The host provides nutrients, the bacteria provide light. In that case, the light isn't produced directly by the animal's tissue, but by colonies of microorganisms housed in specialized chambers with reflective walls and shuttering mechanisms that the animal controls muscularly. The fish opens and closes that chamber to modulate the light signal without altering the bacterial activity itself. This partnership between animal and bacteria represents an alternative solution to developing one's own bioluminescent machinery, outsourcing light production to a specialized organism that already has that process down. Quantum yield, the proportion of luciferin molecules that, upon being oxidized, actually emit a photon, varies between species. The most efficient systems approach a quantum yield of 1. Practically every oxidation reaction generates a photon. This contrasts with artificial photochemical processes, where losses from heat, lateral fluorescence, or parallel reactions significantly reduce the output. This near-maximum efficiency didn't arise by chance. Organisms that waste energy producing heat instead of light need more metabolic resources to sustain their signaling. Over time, more efficient versions of the system were favored because they cost less energy to run. Replicating bioluminescence in the lab became possible decades ago. Purified luciferin and luciferase, mixed in a test tube with oxygen and the right cofactors, produce measurable light. This principle underlies widely used molecular biology assays, quantifying cellular ADP, detecting gene expression, measuring cell viability in culture. Commercial kits containing luciferin and recombinant luciferase are standard items in labs around the world. The reaction that evolved on the ocean floor to allow a shrimp to signal in absolute darkness became, decades after its biochemical characterization, a routine tool in medical and pharmaceutical research, applied in drug screening, infection diagnosis, and cell therapy monitoring. The ability to emit light has been documented in 695 genera of marine animals. To put that number in perspective, a single genus can contain dozens or even hundreds of species. Single-celled bacteria, microscopic dinoflagellates, worms, crustaceans, squid, fish, sharks, jellyfish. Biological light cuts across every major group in the marine animal kingdom. A 2024 study published in the journal Biological Reviews revealed that bioluminescence is more common than previously thought in deep-sea shrimp. More species appear to rely on photophores, light-emitting organs embedded in tissue. A photophore is a light-emitting organ made up of a lens, reflector, and photogenic cells. Some organisms have hundreds of these organs distributed across their bodies, each capable of being activated independently, in specific patterns and sequences, functioning, in certain cases, as a highly complex signaling system. 695 genera is a number that needs context to really land. By comparison, all known terrestrial mammals, lions, whales, bats, rodents, primates, elephants, add up to roughly 500 genera. Marine bioluminescence surpasses the full taxonomic breadth of every mammal on the planet, and that number refers only to what has been formally documented. Estimates suggest that significant portions of the deep ocean remain unexplored. Every expedition with submersible vehicles equipped with sensitive cameras comes back with footage of uncatalogued organisms. The list of 695 genera, therefore, represents a floor, not a ceiling. Bioluminescent sharks caught the scientific community off guard when detailed studies of deep water species revealed light-emitting skin patterns. The southern lantern shark, for example, produces bioluminescence across its ventral surface using photophores distributed throughout the skin. This shark inhabits the mesopelagic zone and uses counterillumination to reduce its silhouette when seen from below against the faint glow that still filters down from above. What looks like passive camouflage is actually an active, energetically costly process. The animal continuously regulates the intensity of its emitted light to match the ambient illumination. The discovery expanded the inventory of bioluminescent vertebrates and showed that the ability is more widespread among fish than previously assumed. Polychaete worms, marine annelids frequently overlooked in biodiversity surveys, include some notable bioluminescent species. The Tomopterus worm emits yellow flashes, a rare color among marine organisms, whose biochemical origin hasn't been fully worked out. The luciferin it uses doesn't match any of the molecular structures known in other groups. This case illustrates a recurring pattern. Phylogenetically distant organisms that independently developed bioluminescence often use different chemistries to arrive at the same optical result. Mapping these different molecular solutions provides insight into the pathways through which bioluminescence has repeatedly emerged throughout the history of marine life. Echinoderms, the group that includes sea stars, sea urchins, and sea cucumbers, have bioluminescence cases that are less studied than those in fish and cephalopods, but equally documented. Brittle stars emit green light when disturbed. Some deep-sea cucumbers release luminescent particles into the water when attacked, a strategy that may disorient predators or attract secondary predators that then turn and threaten the original attacker. This second hypothesis, called burglar alarm signaling or third-level signaling, implies that the organism uses light not just to communicate with its immediate aggressor, but to recruit a third party into the conflict, an indirect defense strategy of considerable sophistication. The taxonomic distribution of bioluminescence among marine organisms isn't random. Groups that predominantly inhabit the mesopelagic and bathypelagic zones show far higher proportions of luminous species than groups restricted to shallow waters. Fish of the family Myctophidae, lanternfish, are one of the most biomass-abundant groups in the ocean and are almost universally bioluminescent. Estimates suggest that lanternfish make up a significant share of vertebrate biomass in the open ocean. The fact that the most abundant animal group in one of Earth's largest habitats is almost entirely bioluminescent gives real weight to the role this ability plays in the ecological structure of the deep oceans. Photophores vary enormously in complexity across species. In the simplest ones, clusters of photogenic cells sit just below the skin with no additional structure. In the most elaborate, the photogenic cell is surrounded by a reflective layer that directs the emitted light, covered by a lens that focuses it, and filtered by a color filter that selects the transmitted wavelength. Some species also have a muscular shutter that controls when the light is released outward. This progression in structural complexity mirrors the diversity of functions bioluminescence serves. The more precise and controllable the signal, the more sophisticated the photophore needed to produce it. Cephalopods, the class that includes squid, octopuses, and nautiluses, boast some of the most elaborate bioluminescent systems. The squid Wattisenia scintillans, found in Japan, has photophores on its tentacles that emit polarized light, a property of light that's rarely exploited biologically. This squid's eyes have photoreceptors sensitive to polarization, allowing it to detect patterns that would be invisible to non-specialized eyes. This communication channel is opaque to predators without that ability. The squid signals to members of its own species without giving away any usable information to surrounding organisms. Polarization functions like a restricted access communication protocol, detectable only by those with the right receiver. The number of independent origins of bioluminescence in the animal kingdom is an active area of research. Phylogenetic analyses, which reconstruct family trees among organisms using genetic data, estimate that bioluminescence arose independently at least 40 times in marine fish and dozens of additional times in other groups. Each independent origin represents a separate experiment by nature converging on the same solution. Comparing the different chemistries, structures, and regulatory mechanisms across these independent origins provides data on which aspects of the system are non-negotiable, present in every solution because there's no viable alternative, and which are contingent, varying from group to group. Genetic sequencing technologies and high-resolution imaging have transformed the pace of discovery in the field. Genes encoding luciferases from different species are identified, compared, and expressed in laboratory systems for functional characterization. Organisms collected on deep-sea expeditions have their genomes sequenced before they're even formally described as new species. This accelerating flow of data continuously expands the map of bioluminescence in the ocean, filling in taxonomic gaps and revealing lineages where the ability was present but hadn't been detected through direct observation methods. The inventory of 695 genera, built over decades of fieldwork and laboratory research, keeps growing with every expedition that descends below 200 meters and comes back up with samples from the most extensive and least understood habitat on Earth. Picture a beach at 11 p.m. The waves roll in, and each one lights up blue, a glow bright enough to illuminate the faces of people standing on the sand. That spectacle is produced by organisms less than half a millimeter long. Dinoflagellates are a type of single-celled algae, and each one of these tiny organisms emits a small amount of light. When conditions are favorable, populations double rapidly. Each cell divides asexually, producing clones. When nutrients run out or temperature drops below a certain threshold, cells form resistant cysts that sink and lie dormant in the sediment until conditions improve. This cycle of blooming and dormancy creates the seasonality observed at bioluminescent beaches. The spectacle doesn't happen year-round in the same spot, concentrating instead in the months when temperature and nutrient availability hit the sweet spot for that specific species. Human disturbance directly affects bioluminescent dinoflagellate ecosystems. Light pollution reduces the visibility of the phenomenon for observers, but doesn't directly interfere with the cell biology. Chemical pollution and eutrophication, excess nutrients from sewage and agricultural fertilizers, may initially boost dinoflagellate growth, but frequently result in the proliferation of non-bioluminescent species competing for the same resources. Salinity changes caused by hydrological modifications, river diversions, canal construction, disrupt the balance that sustains high population densities. Mosquito Bay, designated as a nature reserve, receives formal protection precisely because the integrity of the surrounding ecosystem is a prerequisite for the luminous phenomenon to persist. Casiabata is the local name given to the sea glow in some coastal communities in Southeast Asia, where fishermen learn to read bioluminescence as an indicator of fish school locations. Fish moving through the water activate surrounding dinoflagellates, creating luminous trails visible in the dark that give away the position and direction of the school. This ecological knowledge accumulated over generations connects a cellular biochemical phenomenon to the everyday practice of artisanal fishing. The same light produced by a single cell detecting mechanical deformation in its membrane becomes, when multiplied by billions of organisms in a dark bay, navigable information for boats that have no electronic detection equipment, translating biochemistry into practical guidance within the darkness of the nighttime ocean. At the bottom of the ocean, between 200 and 2,000 meters deep, lives the female anglerfish. Above its head, a flexible rod ends in a structure called a lure, and inside that lure live bacteria that produce light continuously. Most female serratioid anglerfishes carry extracellular symbiotic luminescent bacteria in that lure-shaped projection above the animal's head. The bacteria glow to attract food for the fish, while the fish provides the bacteria with the nutrients they need. The most common bacteria found in the lures belong to the family Bioluminescence, specifically the genera Photobacterium and Vibrio. In the absolute darkness of the deep sea, any light signal can indicate food, a mate, or a point of orientation. A smaller fish spots the point of light and moves toward it. The anglerfish's mouth stays dark and motionless, invisible. When the prey is close enough, the jaw snaps shut in a fraction of a second. The lure that did the attracting was built with living light. The anglerfish's light-producing rod is called the Elysium, Latin for lure or enticement. It's a modification of the first dorsal fin spine, which migrated during embryonic development to the front of the head. At the tip of the Elysium, the bulbous structure containing the bacteria is called the esca, or terminal photophore. The length of the Elysium varies across species. In some it's as long as the animal's entire body. In others it's short and positioned close to the mouth. That variation isn't random. Species that inhabit different depths and pursue different prey have lure geometries tuned to maximize attraction efficiency in the specific environment they occupy. The bacteria inside the lure aren't forcibly trapped. They enter into symbiosis with the host through a process that hasn't been fully worked out yet. Newly hatched anglerfish don't have bacteria in their lure. They're acquired from the ocean environment during early developmental stages. Microscopic channels and pores on the lure's surface allow free-floating bacteria in the water to get inside and colonize the interior of the photophore. Once inside, they multiply, taking advantage of the nutrients provided by the host's tissue. The bacterial density inside a mature lure can reach extraordinarily high values. Estimates point to 10^1 cells per milliliter, a concentration that explains the light intensity produced by such a small structure. Bacteria of the genus Photobacterium produce light continuously, unlike dinoflagellates, which fire in response to mechanical stimulation. These bacteria glow non-stop as long as oxygen and nutrients are available. The control of the light signal is handled by the host, not the bacteria. The anglerfish has muscle and pigment surrounding the lure that work like a shutter. By contracting the pigmented tissue, the animal blocks the light. By relaxing it, it lets the glow through. This control allows the fish to vary the light pattern, pulsing, flashing, varying intensity, without altering the internal bacterial activity. The lure transforms a biologically continuous light source into a signal the fish can modulate based on whatever the hunting situation calls for. The anglerfish's mouth is disproportionately large relative to its body. Long, translucent teeth curved inward prevent prey from escaping after first contact. The stomach is highly distensible. The species Melanocetus johnsonii, with a body of about 16 centimeters, can swallow prey larger than itself thanks to the elasticity of its stomach walls and the loose articulation of the jaw. This ability to consume large prey is an adaptation to the environment. In the deep sea, feeding opportunities are rare and unpredictable. When prey shows up, the organism needs to be equipped to take advantage of it regardless of size. Passing up a meal due to anatomical limitations would be an unacceptable metabolic waste in an environment where energy is a scarce resource. Reproduction in serratioid anglerfish is one of the most unusual systems among vertebrates. Males are drastically smaller than females. In some species, the ratio reaches 60 times in length and hundreds of times in mass. After hatching, the male has a highly developed olfactory system for detecting pheromones released by the female into the water. Upon finding her, he bites into her surface and begins a process of tissue fusion. Circulatory systems progressively connect until the male becomes a permanent parasite, losing his eyes, internal organs, and autonomy. In return, He receives direct nutrition through the shared circulatory system and provides sperm continuously. A single female can carry multiple fused males simultaneously, ensuring fertilization is always available without needing to find a mate again. Different anglerfish species have distinct bioluminescence patterns in their lures, variations in flash frequency, intensity, and color. Researchers propose that these specific patterns may function as species recognition signals, allowing females and males of the same species to locate each other in the dark. In an environment where different serratioid species inhabit overlapping depths without being able to see each other, the ability to distinguish one's own light signals from those of neighboring species would be a considerable advantage. This hypothesis still lacks robust experimental confirmation. Capturing and observing the reproductive behavior of anglerfish under natural conditions remains technically challenging given the extreme environment they inhabit. The relationship between the anglerfish and its luminescent bacteria raises the question of how each host species selects its specific symbiont bacterial species. Genetic sequencing of bacteria extracted from the lures of different anglerfish species revealed a striking correspondence. Certain host species consistently associate with certain bacterial lineages, even when collected from different oceans. This pattern suggests that the symbiont selection process isn't random. The host has molecular recognition mechanisms that favor colonization by specific lineages. Identifying these mechanisms is an active area of research, with implications for understanding how symbiotic partnerships between animals and bacteria get established and maintained over time. Outside the context of the anglerfish, bioluminescent bacteria of the genera Vibrio and Photobacterium occur freely in the ocean, associated with the surfaces of dead animals, the digestive tracts of fish, and coastal sediments. Free-living bacterial bioluminescence is regulated by a mechanism called quorum sensing. The bacteria monitor population density in their surroundings by producing and detecting signaling molecules called autoinducers. Below a certain density threshold, light production is suppressed. Above it, the bioluminescent metabolic pathway is activated across the entire population simultaneously. This system prevents isolated bacteria from wasting energy producing light with no audience. Emission only occurs when there are enough cells to generate a visible and biologically relevant signal. Research using remotely operated underwater vehicles captured anglerfish hunting behavior in the wild for the first time at depths close to 800 meters. The footage shows the animal nearly motionless, with the lure swaying gently in front of its mouth. The lure's movement isn't produced by currents. It's controlled by muscles in the elysium that create a rhythmic oscillation, mimicking the swimming of small organisms. The combination of continuous light with simulated small prey movement amplifies the trap's effectiveness. There isn't just a point of light in the dark, but a point of light that behaves like something alive and vulnerable. This dual simulation, visual and behavioral, turns a fixed anatomical structure into one of the most sophisticated passive predation strategies documented among marine vertebrates. Any animal swimming in the ocean's midwater zone, between 200 and 1,000 meters deep, faces an immediate physical problem. Sunlight still partially penetrates that region, and an animal seen from below looking upward appears as a dark silhouette against the lighter background near the surface, exactly like a bird against a bright sky. To a predator swimming below, that silhouette is an identified meal. Counterillumination is an active camouflage method observed in marine animals, such as the firefly squid and the lanternfish, producing light to match the surrounding environment in both brightness and wavelength. The hatchetfish has downward-pointing light-producing organs and adjusts the amount of light emitted from its underside to match the light coming from above. The predator looks up and sees only the continuous brightness gradient of the water. The silhouette has vanished. Two species of mesopelagic squid significantly alter the color of their bioluminescence during counterillumination, with the color shift triggered by changes in water temperature. These are organisms that calibrate the color and intensity of their emitted light in real time, like an active invisibility system. To understand the problem that counterillumination solves, you need to understand how light behaves underwater. At the surface, sunlight penetrates at maximum intensity with a broad spectral distribution. As depth increases, longer wavelengths, red, orange, are absorbed first. Below 200 meters, the remaining light is almost exclusively blue-green. This selective attenuation creates a gradient. The deeper you go, the less light and the narrower the spectrum. An animal swimming at 300 meters has above it a diffuse blue glow coming from the surface. To a predator below, that bright background makes any opaque object immediately visible as a dark silhouette. Counterillumination solves this problem by actively producing that same blue glow on the animal's ventral surface, eliminating the contrast that would make the silhouette detectable. The hatchetfish, family Sternoptichidae, is one of the organisms that implements counterillumination with the greatest documented precision. Its body is laterally compressed and silver-colored, minimizing the silhouette area seen from the side. On the ventral surface, rows of photophores point directly downward, emitting blue-green light. Additional photoreceptors on the animal's dorsal surface continuously measure the intensity of the light coming from above. That measurement is used as a reference to adjust the intensity of the light emitted by the ventral photophores. When the light from above increases, because the animal moved to a shallower depth or because the time of day changed, the photophores increase their output accordingly. The system works like an automatic real-time brightness control. Measuring whether counterillumination actually works as effective camouflage requires a specific methodology. Researchers developed photometers, light measurement instruments, positioned below tanks containing live hatchetfish, recording the amount of light coming from the animal's ventral surface compared to the background light measured simultaneously. In experiments run under these conditions, the difference between the animal's luminosity and the background water luminosity was less than 0.5%, below the visual discrimination threshold of most predators tested. The camouflage isn't approximate. It's calibrated with a precision that rivals artificially designed control systems engineered by humans. Squid of the genera Abralia and Abraliopsis add an extra layer of complexity to the system. In addition to producing counterillumination, they modulate the polarization of the emitted light. Natural ocean light has a specific polarization pattern determined by the angle of sunlight and the structure of the aquatic medium. Squid in certain groups have polarization-sensitive photoreceptors and adjust their bioluminescent emission parameters to match that ambient polarization. For predators with polarimetric vision, capable of detecting discrepancies in the polarization state, an animal emitting light with the wrong polarization would be just as detectable as one emitting nothing at all. Matching the polarization closes off that additional vulnerability. Water temperature influences the color of bioluminescence in at least 2 species of mesopelagic squid studied in detail. In colder water, the emission shifts slightly toward shorter wavelengths, a more intense blue. In warmer water, the spectrum shifts toward green. This variation tracks the change in ambient light spectrum. Under different ocean conditions, water transparency favors slightly different wavelengths. An animal that calibrates its emission according to local temperature automatically adjusts its camouflage to the optical characteristics of its immediate environment, without needing to directly measure the light. Temperature acts as a proxy, an easily detectable variable that predicts the optical properties of the medium with enough precision to guide the bioluminescent adjustment. Counterillumination isn't limited to the animal kingdom. Aeronautical engineers studied the principle for potential aircraft applications. Illuminating the underside of a wing or fuselage with intensity matching the sky above could reduce visual detectability to the naked eye or to passive optical detection systems. Prototypes were developed during the 20th century with limited success due to the difficulty of rapidly adjusting the emission as altitude, sun angle, and cloud cover changed. Marine organisms solved those same problems through sensors integrated directly into the body and direct nervous control of photophores, a more agile and adaptive solution than any artificial system based on discrete electronics developed to date. Beyond camouflage, counterillumination may serve a secondary function in intraspecific communication. Ventral photophore patterns differ between species and, in some cases, between males and females of the same species. Researchers propose that these specific patterns allow recognition of individuals of the same species at close range, even when the primary function of the emission is camouflage from distant predators. The two functions aren't mutually exclusive. The emitted light simultaneously obscures the silhouette from predators below and signals identity to members of the same species nearby. Using the same resource for two different purposes is a metabolically efficient strategy in an environment where the energy cost of each process is high and feeding opportunities are irregular. Research on counterillumination got a significant boost with the development of hyperspectral cameras, devices capable of capturing images across dozens or hundreds of wavelengths simultaneously, far beyond the 3 bands recorded by conventional cameras. With this equipment, researchers can precisely measure the exact spectrum of light emitted by each region of the animal's body and compare it to the ambient spectrum measured simultaneously. Tiny discrepancies that would go unnoticed in conventional photography become detectable. Data collected with this technology revealed that the spectral adjustment performed by hatchetfish and mesopelagic squid is more precise than previous estimates had indicated, suggesting that the selective pressure to reduce optical discrepancies was consistent and intense throughout the time these organisms have inhabited the mesopelagic zone. The full set of mechanisms that make up counterillumination, measuring ambient light, adjusting intensity, spectral modulation, polarization matching in some species, represents a distributed control system integrated into the animal's own body. There's no central brain coordinating each photophore individually. Control is partly local, with each part of the system responding to nerve and hormonal signals that translate environmental readings into emission adjustments. This type of distributed architecture, where sensors and actuators are locally coupled without depending on central coordination for every decision, is a principle that robotics and autonomous systems engineers are working to replicate in machines that need to respond quickly to variable environments, and one that marine organisms implemented hundreds of millions of years ago on the dimly lit floor of the open ocean. In some cases, bioluminescence works as a distress signal, and the mechanism is so specific that researchers call it the bioluminescent burglar alarm. A tiny dinoflagellate being swallowed by a copepod, a microscopic crustacean, fires a flash of light at the moment of the attack. That flash attracts the copepod's predator. When the microorganisms light up, they draw attention, what marine biologist Edith Witter calls the bioluminescent burglar alarm. Scientists believe dinoflagellates may be counting on the glow to scare off predators or pull in larger predators that will then eat their pursuers. The vampire squid, Vampirotuthis infernalis, applies a more active version of the same principle. Instead of dark ink, it ejects sticky bioluminescent mucus, which can startle, confuse, and slow down predators, giving the squid a chance to escape. In that context, light functions as a tool for tactical confusion. A copepod is a microscopic crustacean, smaller than a grain of rice, and one of the most numerically abundant animals on the planet. In the ocean, It feeds on phytoplankton and dinoflagellates. When it captures a dinoflagellate cell and begins ingesting it, the mechanical pressure on the cell membrane triggers the bioluminescent reaction. The resulting flash lasts a fraction of a second, but occurs in an environment of total darkness. To any visual predator in the vicinity, that point of light signals activity, and activity in the deep ocean often means available prey. The copepod, by attacking the dinoflagellate, unwittingly gives away its own position. The organism being attacked has no nervous system, makes no conscious decision, plans no response. The light is an automatic consequence of a physical stimulus converted into a chemical reaction. Marine biologist Edith Witter dedicated decades to studying ocean bioluminescence and developed observation instruments capable of recording faint light emissions at great depths without disturbing the surrounding organisms. Ultra-low-light cameras coupled to silent submersible vehicles made it possible to observe behaviors that conventional systems masked. The propulsion lights and white illumination of submarines disturb sensitive organisms and alter natural behavior. Witter documented that a large portion of ocean bioluminescence occurs in response to mechanical disturbance, and that the chain of attraction, dinoflagellate lights up copepod, copepod draws in a larger predator, can extend across several trophic levels, with each link in the food chain potentially revealed by the glow of the previous one. The vampire squid, Vampirotuthis infernalis, inhabits the oxygen minimum zone, a region between 600 and 900 meters, where dissolved oxygen concentrations drop to levels most predators can't tolerate. That hostile environment offers refuge. Few pursuers can operate there. When threatened outside that zone, or when a predator adapted to low oxygen finds it, the vampire squid has a defensive arsenal that includes bioluminescent mucus. Unlike octopuses that squirt opaque ink to create a visual blackout curtain, the vampire squid ejects a secretion that glows intensely. The effect is the opposite. Instead of darkening, it lights up, possibly overwhelming the attacker's senses or attracting a third predator that then becomes a threat to the original pursuer. Bioluminescent mucus as a defensive weapon is a rare solution among cephalopods. The chemical makeup of the mucus ejected by the vampire squid includes luminescent particles that keep glowing for minutes after ejection, unlike the instantaneous flashes produced by dinoflagellates or by nerve-controlled photophores. That prolonged glow in the mucus serves a distinct purpose. It's not a quick signal that draws attention to a single point, but a persistent distraction that stays active while the animal flees. Predators with visual systems sensitive to movement may lock onto the residual glow floating in the water, losing track of the real animal's escape route. In this case, light functions as an evasion decoy, a fake object left behind while the real organism vanishes into the darkness. The biological alarm principle, using light to recruit third parties against an aggressor, shows up in multiple ecological contexts beyond the ocean. Terrestrial plants release volatile compounds when attacked by herbivores, attracting parasitoids that then go after the herbivores. Some aphid species release alarm pheromones when touched by predators, alerting nearby individuals. Marine bioluminescence as an alarm represents an optical version of that same general principle, signaling that uses the surrounding environment as an amplifier to turn an individual attack into an event detectable by third parties. The difference is the channel, light instead of chemical compounds or mechanical vibrations, selected by evolutionary pressure in an environment where visibility is zero and self-produced light is the only signal that travels distances without being lost to currents or chemical diffusion. Sessile organisms, those fixed to a substrate, unable to flee, rely entirely on defenses that don't require mobility. Cnidarians such as corals and anemones have bioluminescent representatives that emit light when mechanically disturbed. The defensive function of bioluminescence in sessile organisms is debated. Without the ability to escape, attracting secondary predators would be the most plausible strategy, but direct experimental evidence is hard to come by. An alternative proposal is that the light serves as an intraspecific stress signal. Nearby organisms of the same species, upon detecting a glow in their neighbors, might ramp up production of chemical defensive compounds or contract their tissue before the disturbance reaches them, reducing damage through anticipation. A single dinoflagellate flash carries negligible energy, not enough to be seen at any meaningful distance. The effectiveness of the biological alarm depends on population density. At low concentrations, scattered individual flashes produce no coherent detectable signal. At high concentrations, cascade activation. One flash triggers mechanical disturbance in the water that activates neighboring cells, amplifies the original signal. The same initial event that would produce imperceptible light from an isolated cell generates, in a dense population, a wave of glow radiating outward from the point of attack. This collective amplification transforms an individual cellular response into an ecologically relevant signal, detectable by predators at distances where a single flash would be invisible, converting population density into a collective defensive advantage. Researchers developed a camera system called SPLAT, splash light from amplified turbulence, to quantify bioluminescence in open water without capturing organisms. The equipment records light emitted by organisms disturbed by a controlled jet of water, making it possible to estimate the density and luminous capacity of the populations present. Expeditions using this method revealed that bioluminescence is detectable at virtually every sampled point in the open ocean above 1,000 meters, regardless of geographic location. The ubiquity of the phenomenon suggests that the ecological pressures favoring the biological alarm are globally distributed. The entire ocean operates, to some degree, as a system where light produced by small organisms in response to attack is a functional part of the food chain. The chain connecting dinoflagellate, copepod, and secondary predator illustrates how bioluminescence is woven into ocean ecology at multiple levels simultaneously. The flash from a single attacked organism doesn't just affect the immediate attacker. It potentially reshapes predator behavior across a wider area, redistributing who pursues whom for as long as the signal remains active. In waters densely populated with bioluminescent organisms, successive attacks generate linked signals that create a dynamic luminous map of local predatory activity. That map is readable by any animal with vision sensitive to blue light, and in the mesopelagic ocean, that includes most active predators. The light manufactured by microscopic organisms in response to mechanical pressure on their membrane becomes, at ecological scale, shared environmental information that structures hunting and escape behavior in one of the most densely inhabited regions on Earth. In 2021, scientists discovered 3 new species of deep-water sharks that produce bioluminescence through specialized cells in their skin. The southern lantern shark, *Etmopterus granulosus*, was one of the species studied. It has thousands of photophores distributed across its belly, producing a soft, continuous blue-green glow. The researchers' hypothesis is that this glow serves for counterillumination, erasing the animal's silhouette against the faint light filtering from above, and possibly for communication between members of the same species in a near-total darkness environment. The general rule in the ocean is that what glows continuously attracts, and what flashes repels. Lantern sharks appear to use continuous luminescence in a more understated way, as a simultaneous identification and camouflage system, operating below the detection threshold of larger predators. Sharks are cartilaginous fish. Their skeleton is made of cartilage rather than bone, and they make up a group of roughly 500 described species, most of which inhabit shallow or mid-depth waters. Deepwater species are significantly less studied for logistical reasons. Collecting live animals from hundreds of meters below the surface without damaging sensitive tissue and without altering the pressure and temperature conditions that keep biological structures intact is technically challenging. The study published in 2021 in the journal iScience was based on specimens collected off the coast of New Zealand and kept temporarily under low-light conditions that allowed researchers to photograph and measure the bioluminescent emission before the animals died from capture stress. The family Atmopteridae, the lantern sharks, includes roughly 40 species and represents the most diverse and best documented group of bioluminescent sharks. All studied members have ventral photophores, but the density, distribution, and pattern of these organs vary between species. In Atmopterus granulosus, photophores are concentrated in specific regions of the flank and belly, forming patterns that differ between males and females. This sexual difference in photophore distribution suggests that part of the bioluminescent function is tied to mate recognition. At depths where visibility is zero beyond a few centimeters, identifying sex and species by light pattern would be a considerable reproductive advantage, especially for animals that inhabit the open ocean with no fixed territories to concentrate encounters between individuals. Light-producing cells in lantern sharks differ structurally from the photophores found in bony fish like the hatchetfish. Instead of cavities surrounded by reflectors and lenses, the photophores of Atmopterids consist of individual cells, photocytes, distributed in the dermis and controlled by hormones rather than direct nerve signals. Melatonin, a hormone produced by the pineal gland that's also present in humans where it regulates sleep cycles, was identified as a suppressor of bioluminescence in these sharks. Prolactin, another hormone, stimulates emission. This hormonal control produces slower variations than direct nerve control, suited for maintaining a stable, continuous glow rather than rapid flashes, consistent with the passive camouflage function that counterillumination requires. The luminous kitefin shark, Deilechias licha, is the largest known bioluminescent shark, reaching up to 180 centimeters. It inhabits depths between 200 and 600 meters and was only confirmed as bioluminescent in 2021, even though it belongs to a genus that has been studied for decades. The delay in confirmation illustrates the methodological challenge. The bioluminescent emission of these animals is faint, visible only in total darkness with sensitive cameras. Labs without the right equipment simply didn't detect the phenomenon. As ultra-low-light cameras and protocols for keeping specimens in controlled darkness advanced, detections that previously slipped through became routine, suggesting that the number of bioluminescent species among deep-water sharks may be substantially higher than the current catalog indicates. Continuous bioluminescence represents a permanent metabolic cost. Keeping photocytes active, synthesizing luciferin, sustaining luciferase enzymatic activity. All of these processes consume energy that could otherwise go toward growth, reproduction, or locomotion. The fact that lantern sharks maintain this cost throughout their adult lives indicates that the benefit, camouflage, interspecific communication, or both, outweighs the energy investment. In environments with low prey density and predators present, reducing the probability of being detected by a larger predator carries enough survival value to justify an ongoing metabolic cost. Bioluminescence in these animals isn't an episodic response to threats. It's a baseline state maintained indefinitely as permanent insurance against visual detection. The discovery of bioluminescence in 3 shark species simultaneously in 2021 didn't result from an expedition specifically planned for that purpose. The specimens were collected as part of a marine biodiversity survey in deep New Zealand waters. Researcher Jerome Malafet from the Marine Biology Laboratory at the Catholic University of Louvain spotted the light emission after keeping the captured animals in a dark tank as a standard analytical procedure. The accidental discovery reflects a common pattern in deep-sea science. Logistical limitations mean data is often collected for one purpose and ends up revealing unexpected phenomena. Estimating how many other deep-water shark species have bioluminescence that's gone undetected due to a lack of adequate observation protocols remains an open question. Intraspecific communication through bioluminescence in sharks implies that these animals have vision capable of detecting faint emissions from members of their own species. Retinal analysis of Etmopterus spinax, a species related to the southern lantern shark, revealed a retina dominated by rod cells, photoreceptors sensitive to low light intensities, with peak spectral sensitivity in the blue-green range, exactly the wavelength emitted by the species' ventral photophores. This match between emission spectrum and peak receptor sensitivity isn't a coincidence. It represents co-adaptation between the light production and light detection systems, shaped over time to maximize the efficiency of communication in the specific environment where these animals live. Photophore patterns as an intraspecific identification code raises the question of whether lantern sharks recognize individuals or only categories: species, sex, reproductive stage. Direct experiments with live animals are difficult to carry out given the capture and maintenance conditions. Computational models based on photophore distribution data and estimated visual sensitivity suggest that the visual resolution of these animals at close range would be sufficient to distinguish patterns between different individuals, but insufficient for individual recognition at greater distances. In this model, bioluminescence would function as group identity at long range and potentially as individual identity at close contact distances. Two levels of information transmitted by the same physical signal in different contexts. The catalog of bioluminescent vertebrates has expanded considerably over the past decade. Amphibians, sea turtles, and certain reef fish species have been added to lists previously dominated by deep-sea organisms. In sharks, the frontier of knowledge is still being established. Of the roughly 500 described species, fewer than 24 have been formally examined for bioluminescence with adequate methodology. The rest remain unevaluated, not confirmed as non-luminescent, simply untested. Every new deep-water expedition with low-light observation protocols has the potential to add species to the list. The shark glowing softly in the abyss, invisible to the naked eye under conventional lighting, awaits cataloging in an ocean whose depths still hold a significant proportion of their diversity without name or formal description. Bioluminescence represents a promising frontier in medical research and diagnostics. The story starts with Equoria victoria, a transparent jellyfish from the North Pacific that produces blue light internally and converts it to green through another protein. That second protein, GFP, green fluorescent protein, was isolated in the lab and began functioning as a molecular tag. It's possible to insert the GFP gene into any cell and make that cell glow when viewed under a microscope. The 2008 Nobel Prize in Chemistry was awarded precisely for that development. Today, the most direct application is in cancer research. Bioluminescent markers help visualize and track tumor growth and metastasis in living animals, allowing researchers to monitor disease progression and evaluate treatment effectiveness in real time. Bioluminescence imaging, BLI, quantitatively monitors biomolecular reactions and cellular behaviors in living organisms using luciferase enzymes with no need for external light. What researchers learned by observing organisms from the ocean floor is being applied to detect and monitor tumors in human patients. Equoria victoria was extensively collected in the waters of Puget Sound in the Pacific Northwest, starting in the 1960s by researcher Osamu Shimomura. Shimomura wasn't looking for medical applications. His question was purely biochemical. How does this jellyfish produce green light if the luciferin-luciferase reaction generates blue light. The answer required isolating the protein responsible for the conversion. After purifying hundreds of kilograms of jellyfish tissue over the course of years, Shimomura identified GFP, a protein that absorbs blue light and re-emits it as green through fluorescence. The discovery remained of limited interest for decades until Martin Chalfie demonstrated, in 1994, that the GFP gene could be inserted into living organisms and express functional protein in cells from species completely different from the original jellyfish. Inserting a gene into a living cell and having that cell produce functional protein is a molecular biology procedure called genetic transformation. The GFP gene, isolated from the jellyfish genome, is connected to the gene for another protein of interest, for example, a protein present only in cancer cells. When this genetic package is introduced into an experimental cell, the cell produces both proteins simultaneously, the protein of interest and the GFP attached to it. Wherever the protein of interest shows up inside the cell, GFP shows up with it and glows when illuminated with blue light. Researchers can thus map, in real time and with nanometer precision, where a specific protein is located, how it moves, and what it interacts with inside living cells. Information that was impossible to obtain with previous techniques. GFP has limitations for in vivo applications, inside complete organisms rather than isolated cells. It requires external blue light illumination to emit. That light penetrates biological tissue only a few millimeters before being absorbed and scattered. For visualizing structures deeper than the surface, fluorescence doesn't cut it. Luciferase-based bioluminescence solves that problem. The chemical reaction generates light internally without needing an external source. Tumor cells tagged with luciferase, implanted in an experimental animal, produce light that passes through enough tissue to be detected by a sensitive camera positioned externally. The tumor becomes an autonomous light source, localizable and quantifiable without opening the animal, without anesthesia, without interrupting the natural progression of the disease being studied. Bioluminescence imaging, BLI, has become a standard tool in preclinical pharmacology. When researchers develop a potential cancer drug, they need to test whether the compound actually reduces tumor growth in a living organism before moving on to human clinical trials. With BLI, the drug is administered to the animal, and the amount of light emitted by the tagged tumor cells is periodically measured. An increase in emission indicates tumor proliferation. A decrease indicates treatment response. The same animal can be monitored repeatedly over weeks without surgical procedures, reducing the number of animals needed in each experiment and improving data quality by eliminating variability between different individuals. The jellyfish glowing in the North Pacific became, through this chain of discoveries, a central instrument in oncology drug development. GFP variants with different emission spectra were developed through protein engineering. Controlled changes in the amino acid sequence that modify the molecule's optical properties. Yellow, cyan, red, and other GFP variants make it possible to simultaneously tag multiple distinct proteins in different colors, observing their interactions in real time in the same microscopic field. This molecular color palette, derived from a single jellyfish protein, made possible experiments that previously required chemical fixation of cells, a procedure that kills them and freezes them at a single instant, losing all the dynamics. With multicolored fluorescent markers, processes like cell division, vesicle transport, and cell-to-cell signaling can be filmed in living organisms with unprecedented temporal and spatial resolution. Luciferases from different species have been cloned and optimized for biomedical applications. Nanoluc, a luciferase derived from a deep-sea shrimp, is 10 times brighter than the firefly luciferase commonly used in the lab and much smaller in molecular mass. a property that makes it easier to fuse to other proteins without disrupting their function. BRET, bioluminescence resonance energy transfer, combines luciferase with a fluorescent protein to measure protein interactions inside living cells. When two proteins tagged respectively with luciferase and a fluorophore come physically close to each other, energy transfers from one to the other and the optical signal changes in a detectable way. This system allows detection of transient protein interactions with millisecond temporal resolution in intact cells. Clinical diagnostics are beginning to incorporate bioluminescence directly. Luciferase-based assays detect bacterial contamination in blood and urine with greater sensitivity than conventional culture methods. Live bacteria carry ADP, which fuels the firefly luciferase reaction and generates a light signal proportional to the number of cells present. Response time drops from days to minutes. In blood transfusions, verifying the sterility of stored blood bags using an ADP luciferase assay is a procedure adopted at some hematology centers. Research is advancing toward detection of viruses and biomarkers of inflammatory diseases using luciferase variants coupled to antibodies that recognize specific target molecules. The same basic principle of the lure and the flash, now converted into a molecular precision diagnostic tool. The path from Equoria Victoria to the oncology lab spanned decades and involved researchers on different continents pursuing seemingly unrelated questions. Shimomura wanted to understand jellyfish biochemistry. Chalfie wanted tools for cell biology. Roger Chin wanted to expand the color palette available for microscopy. None of the three deliberately designed a tool for tracking cancer. The development emerged from the accumulation of basic discoveries connected retrospectively by applied researchers who recognized the potential. That trajectory is representative of how fundamental science, research without an immediate applied objective, often generates the most transformative instruments for subsequent generations, on a timescale that no short-term planning could anticipate or directly fund. Beyond cancer, BLI is applied to the study of infections, neurodegenerative diseases, and immunological processes. Pathogenic bacteria tagged with luciferase are introduced into experimental animals, and their spread through the organism monitored in real time. Researchers track how an infection in one organ disseminates to others, testing antibiotics and identifying more effective intervention windows. Neurons tagged with luciferase make it possible to track cell death in Alzheimer's and Parkinson's models, correlating loss of bioluminescent signal with the progression of behavioral symptoms in the same animal over time. Tagged immune cells reveal how the body's defense system responds to vaccines and adoptive cell therapies. Technology derived from a jellyfish, illuminating with unprecedented precision the internal mechanisms of diseases that affect hundreds of millions of people. 76% of individuals observed throughout the entire water column have bioluminescent capability. More than 97% of cnidarians were bioluminescent, and 9 of the 13 taxonomic groups evaluated were dominated by bioluminescent organisms. Throughout most of the ocean, at any sampled depth, the majority of animals around an observer are equipped to produce light. Jellyfish, shrimp, squid, fish, worms, all carrying the same fundamental chemical reaction. Luciferin plus luciferase plus oxygen equals photon. Each organism uses that light differently, to attract, to camouflage, to communicate, to escape, to hunt. Below 200 meters, where sunlight disappears, light production is decentralized and constant, manufactured cell by cell, by trillions of organisms that have never seen the sun. From microscopic single-celled algae to the Humboldt giant squid, ocean animals have adapted the use of light in wildly different ways. The reaction that starts with luciferin and luciferase in a half-millimeter dinoflagellate is the same one that lights the anglerfish's lure in the abyss, erases a lantern shark's silhouette, and tags tumor cells in an oncology lab. The number 76% deserves some context. In a tropical rainforest, the terrestrial habitat with the greatest known biodiversity, the proportion of animals capable of producing biological light is close to zero. Fireflies and a handful of other isolated representatives. In the deep ocean, the situation is reversed. Producing light is the norm, not the exception. That contrast reflects a fundamental difference in the ecological pressures of the two environments. In the forest, vision based on sunlight is dominant, and producing one's own light offers only a marginal advantage. In the mesopelagic and bathypelagic ocean, the complete absence of sunlight turns any biologically produced photon into a high-value resource, whether as a signal, camouflage, or a trap. Where ambient light disappears, manufacturing it internally becomes such an advantageous solution that it was independently adopted by nearly every major marine animal group. Cnidarians, the phylum that includes jellyfish, anemones, corals, and hydroids, show a bioluminescence rate exceeding 97% among sampled individuals. That near total value indicates that luminescent capability arose early in the group's history and was conserved over hundreds of millions of years without significant loss. Bioluminescent jellyfish are frequently transparent. A gelatinous, unpigmented body allows internally produced light to escape without absorption. When disturbed, some species light up completely, turning a translucent structure into an object visible at a considerable distance in absolute darkness. The body's transparency, which in a lit environment would make the animal invisible by eliminating any opaque surface, combines with bioluminescence to create a toggle between passive invisibility and active signaling as the situation demands. Taxonomic groups dominated by bioluminescent organisms include not only cnidarians and mesopelagic fish, but also siphonophores, colonies of highly specialized organisms that coordinate functions among distinct individuals as if they were a single being. Fissionect siphonophory, a group that includes some of the longest animal structures in the ocean, has zooids, individual colony units, specialized for light production. Attacks on these colonies trigger coordinated bioluminescence along the entire structure, potentially illuminating lengths of several meters simultaneously. That coordinated response to localized disturbance implies communication between zooids, a mechanism not yet fully worked out at the molecular level, but one that results in one of the most spectacular manifestations of bioluminescence in colonial organisms. The uniformity of the bioluminescent proportion throughout the water column, from sea level down to 3,600 meters, was an unexpected result of systematic quantitative studies. Prior expectation held that bioluminescence would be more concentrated at depth, where the absence of sunlight would make the phenomenon most advantageous. Data collected with sampling nets and submersible vehicles showed a remarkably stable proportion at all depths. The prevailing interpretation is that the ecological pressures favoring bioluminescence, visual predation, intraspecific communication, camouflage, are present in roughly equal measure throughout the entire water column, with different functions becoming more or less relevant depending on depth, but the total benefit remaining high enough to sustain the capability across virtually all marine animal groups at any sampled depth. The Humboldt squid, Dosidicus gigas, reaches 2 meters in length and 50 kilograms in weight, making it one of the largest bioluminescent cephalopods. It inhabits cold currents in the eastern Pacific, between 200 and 700 meters deep during the day, rising to the surface at night to feed. Its photophores cover virtually the entire body surface and produce rapid, coordinated flashes. Observations by divers and submersible vehicles have documented that groups of Humboldt squid synchronize light pulses during cooperative hunts, flashes propagating through the group in a coordinated way. possibly guiding the positioning of individuals relative to prey or relative to each other. If confirmed as intentional communication, this bioluminescence-mediated cooperative behavior would represent one of the most sophisticated uses of biological light documented in cephalopods. The deep ocean is Earth's largest continuous habitat by volume. It's estimated that roughly 95% of the planet's habitable volume corresponds to marine waters deeper than 200 meters. The zone where bioluminescence dominates as a form of light-based communication. Earth's entire land surface, including all forest, savanna, desert, and tundra ecosystems, represents a small fraction of the total habitat volume available in the biosphere. That scale puts bioluminescence in an ecological position that rarely gets the recognition it deserves. The dominant communication mechanism in the planet's largest habitat is the biological manufacture of light, not the reflection of sunlight. What strikes human observers as an exotic and rare phenomenon is, on a planetary scale, the most widely distributed signaling process among animals. Trillions of organisms continuously producing light in the deep ocean represent a phenomenon whose scale is hard to wrap your head around. Each individual consumes a minuscule amount of energy in the bioluminescent reaction, but the sum of trillions of simultaneous reactions across the full extent of the global ocean constitutes a biologically significant energy flow. Researchers have attempted to estimate the total luminosity produced by the bioluminescent ocean. The uncertainties are enormous given the number of variables involved: varying population density, the proportion of organisms active at any given moment, emission efficiency per species. Even with wide margins of error, the estimates confirm that the deep ocean isn't dark in any absolute sense. It's continuously dotted with flashes and glows produced by organisms that have never come into contact with sunlight and build their own illumination from chemical reactions inherited from ancestors who discovered, hundreds of millions of years ago, that luciferin plus luciferase plus oxygen generates a photon, and that a photon, in absolute darkness, is everything. The story that began with a beach glowing at midnight and a question about how microscopic organisms manufacture light traveled through dinoflagellates that fire at the slightest touch, bacteria glowing inside the lures of abyssal fish, sharks that erase themselves from predators using their own light, and jellyfish that provided the protein now tracking tumors in oncology labs. The chemical reaction underlying all of these phenomena is the same: controlled oxidation of luciferin, catalyzed by luciferase, releasing energy as a visible photon. What varies is the organism carrying it out, the structure housing it, the mechanism controlling it, and the ecological purpose it serves. In a habitat that makes up the largest share of Earth's habitable volume, this fundamental biochemical reaction is the dominant language of communication, and it still holds, in the uncatalogued species and unobserved behaviors of the deep ocean, an unknown proportion of variations on the same luminous theme that science has yet to access and describe.